I'm Feng-Sheng Hu. I'm the Dean of the College of Liberal Arts and Sciences. And I'm also a professor in the Department of Geology and the Department of Plant Biology. I'm just so delighted to see so many people here. It's got to be the popularity and the stellar reputation of Professor Jim Robinson. So thank you all for coming to the inaugural Dean's Distinguished Lecture. The College of LAS is home to an amazing array of scholars who do groundbreaking research, create important understanding of people, culture, language, and literature, and further our knowledge in many, many important areas. We have more than 600 faculty members, many of whom are leaders in their fields, and many of them get invited to give speeches all over the world. We have about 15,000 students this year in the college. They have the opportunities to learn in the classroom from these faculty members, and so do students from all over the campus. But we want to provide ample opportunity for everyone to hear from these outstanding scholars. The College of LAS created this new lecture series to make sure that our campus community has the opportunity to hear from our leading experts about the work they and their students are pursuing to address important questions and improve the society and the world. I'm pleased to introduce our inaugural Dean's Distinguished Lecture Speaker, my colleague, Jim Robinson. Professor Robinson is the director of the Woese Institute for Genomic Biology. He is a Center for Advanced Study professor of entomology, and he holds a Swanlund chair. Jing is a professor in entomology in the School of Integrated Biology, and also holds appointments in the Department of Cell and Developmental Biology, the Program in Ecology, Evolution, and Conservation Biology, and the Beckman Institute. Jing has been at Illinois since 1989. He received his PhD from Cornell University and was an NSF postdoctoral fellow at Ohio State University. He pioneered the application of genomics to the study of social behavior, led the effort to sequence the honeybee genome, and authored and co-authored over 300 publications. He has trained numerous postdocs and students, graduate students. About half of them are now professors at various institutions all over the world. Professor Robinson is one of the masterminds of the BioGenome Project. The project has 3 goals: protect biodiversity, better understand ecosystems, and benefit human welfare. Jing always sets ambitious goals, and the BioGenome Project is no exception. It strives to sequence the DNA of all life on Earth in the next 10 years. This is truly an incredibly ambitious goal, and I know Jing well enough that I would not bet against him. Jing, please come up to the podium.
Thank you very much for that kind introduction. Everyone hear me okay in the back? Great. Well, it's really an honor to be the inaugural speaker, and it's just a real pleasure and an honor to see so many of you coming out for this talk today. This will not be a data-driven talk. The idea is pretty simple. It's already there, captured beautifully in Feng-Sheng's introduction. So I want to flesh out the idea for you, discuss it with you, some of the issues, and have a conversation with you about the idea. I've prepared about maybe 35, 40 minutes of material, so there'll be plenty of time for us to discuss this. The project has a distinct Illini flavor to it. It was conceived by the 3 individuals that you see on the slide. Harris Lewin, a longtime faculty member here in the Department of Animal Sciences and the founding director of the Woese Institute for Genomic Biology, the IGB, and then John Kress at the Smithsonian, and myself. Just by way of brief background, we at the IGB started a plan for a collaboration with the Smithsonian. They had made plans to sequence tens to hundreds of thousands of individuals, individual species, the genomes of tens to hundreds of thousands of individual species. And we started talking with them about how campus in general and how the IGB in particular might be able to collaborate on the analyses of these genomes, introducing them to the Carver Biotechnology Center, which has a national reputation for genome sequencing, assembly, and so forth. And out of that conversation, we started to think, well, maybe hundreds to hundreds of thousands of species might not be thinking big enough. And independent of that, Harris was also thinking along those lines. So we decided to join forces and launch, or at least conceive, of the Earth BioGenome Project. So today what I'd like to do is talk about the motivation for the project, the plan that we have, and then our progress to date. So with that, what is the Earth BioGenome Project? Here it is laid out for you. It's a consortium of independent partner institutions that have the common goal of sequencing and annotating the genomes of all 1.5 million or so described species of eukaryotes. These are animals, plants, and unicellular organisms that are eukaryote, which means they have a special compartment where they keep their genetic material. And that's different than the bacteria and the archaea. And so that distinguishes one way of classifying life. We decided to put the microbes, keep them off of this project because there already is a national microbiome initiative that was started in the Obama administration, which has some momentum. So we decided to focus on the eukaryotes. You'll notice a lot of the language of the project, though, talks about sequencing life. It's a way of speaking a little more easily, but I'm here with friends, and so I'm sharing with you that little bit. So the idea is to sequence the DNA of life in 10 years. And what we see this is as a foundation for science, a new infrastructure for biology, both in the conservation context and in the context of new discoveries. So what are some of the motivations? So here's in case I forgot to say what I just said before, I have it right here. So as many of you know, Illinois is a powerhouse in microbiology, the Department of Microbiology, other departments on campus. We're privileged to have many microbiologists work at the IGB. So I wanted to make sure they knew about that distinction that we are making. So some of the motivations, this quote from E.O. Wilson: "The sixth great extinction spasm of geological time is upon us." There are some 20,000 endangered species. And so we feel some urgency for this project, at least to preserve the DNA of extant species, because they're going away as we speak. Here is a perhaps little known fact. So you read the paper, genomics is everywhere, it's a machine, it's big, it's exciting, but it's only been going on 30 years, and it's still a very new science, and only 0.3% of the species, eukaryotic species, have been sequenced, have had their genomes sequenced to date. Only 0.3%. That is all information based on genomic information, all knowledge in biology based on genomic information comes from a vanishingly small number of species. And so another way to think of this is think of all the discoveries that could be made if we had the other 99.7% species there. And so that's the goal. So why do we want to do this? Well, Fangsheng articulated some of the points: conserve, protect, and restore biodiversity, to revolutionize biology, and to create new benefits for society and human welfare. Why do we think genomes are able to do this? It's because of what I have on the slide here. Genomes are basically the ultimate book of life. So what do I mean by that? You can read the genome as a history book. It contains the evolutionary history of relationships between species. It contains the history of the adaptations, the evolutionary changes that species have made over geological time to be able to adapt to changing conditions. So we can read it as a history book, contains a lot of really great nuggets of history. What's a nugget of history? Well, here's one for you. It had long been thought that Neanderthals and our species, Homo sapiens, kind of kept very separate, and Neanderthals had a mysterious demise. We really didn't know what happened to them. We now know, thanks to genomes, reading ancient genomes in this case, that Neanderthals and our ancestors got together on Saturday nights, maybe Friday nights also, and there is a good record of Neanderthal DNA in human genomes. And it's somewhat of a parlor game now to think about how much Neanderthal DNA is in your spouse's genome or in your friend's genome on a bad day. And so we've really changed our understanding of history with respect to that because of genomes. So it's a history book, works very well as a history book. It also works very well in the present tense as a parts manual. What parts are in an organism? How do they work? What are the biological processes that underlie the functioning of a brain, the ability of a brain to regenerate, to learn, the ability of a plant to adapt to changes in sunlight and alter what it's doing? All of that is contained in the motif of the genome as a parts list. Composing and comprising all of the components or encoding all of the components in organisms. And finally, we can also read it as a prophecy. It contains nuggets that help us predict the future, help us predict risk of disease for humans, help us predict what species of organisms are going to have an easier or a harder time to adapt to climate change. And so it's all there. Now, it's all there in a very coarse form. Genomes contain information that's very far away from the kinds of knowledge that we want. And so there are a lot of steps that go from getting a genome sequence, reading the genome sequence, to obtaining knowledge from it. But it starts there. That's why we call it an infrastructure. It's not the end game. It's just the beginning of the revolution, to have that kind of information. So I want to make sure for the whole audience that everyone appreciates what goes into sequencing a genome, because I'll be referring in different ways to that. So you start with a genome. The human genome, it has 3 billion nucleotides, individual chemical components. It's about 6 feet long, and we have 2 copies coiled up in every one of our cells, and that comprises the genome. So the way we sequence a genome right now is we take that 6-foot-long piece of DNA, and we have enzymes that have been discovered in molecular biology to cut the genome in particular ways. We cut it into hundreds of thousands of pieces, and then we sequence the genomes of those pieces. So we sequence the DNA that's in those pieces, and then we now have a tremendously complicated puzzle composed of hundreds of thousands or millions of pieces. And then the biologist turns to our smart computer science friends and say, we're done. Now, can you put this really complicated puzzle back together again? If you told the story to a 6-year-old, they would say, boy, that's really a dumb way to do it. Why don't you just take the 6-foot-long molecule and read it from the beginning to the end? Here you are shooting your mouth off that it's a book, so read it from the beginning, middle, to the end. And that's the goal, of course, but we're not there yet. So we are in the 3rd generation of sequencing technology, and the sequencing technology marches forward by reading ever longer pieces of DNA. So eventually we can hope for the scenario that I just described, but for now we are quite a ways away from that. We're in our 3rd generation. So with that as an interlude, back to the motivations for the project. So here's in a little more detail: maximize returns to society and human welfare, developing new treatments for infectious and inherited diseases, creating new biosynthetic fuels, generating new biomaterials, generating new approaches to feeding the world, and identifying drugs to slow or reverse aging. And I'll just wear my IGB hat and say with great pride that, with exception of the aging, we have research going on at the IGB, cutting-edge research that uses genomes to address 4 of those 5 right now, based on 0.3% of the species. We want to revise and reinvigorate our understanding of biology and evolution. What you see here is a graphic that depicts the tree of life the evolutionary relationships between organisms, between species, excuse me. And history has told us, recent history has told us that when you have additional genome sequences and put them into play, you can understand better evolutionary relationships. And so there are many cases, and there are many leading practitioners of this work in the room here, where you use genetic information from genomes to be able to revise your understanding of what species is related to what species. That's foundational for all of biology, to know that kind of information. Who's older, who begat whom, and what the relationships are. And genomes provide that information. Changing gears, and something of increasing interest are both of these topics, the bioeconomy, infrastructure, and enhancing biodiversity. So there's a big emphasis in the current administration on bioeconomy. There was a bioeconomy summit just a couple weeks ago. Our VCR, Susan Martinez, was one of only 2 VCRs, Vice Chancellors for Research, invited to participate in the President's Summit on the bioeconomy. We are in discussions with the Office of Science and Technology Policy. I'm going there Friday again for another meeting to talk about the role of Earth BioGenome Project, EBP, in the bioeconomy. So it's something of increasing interest, as is, and no surprise, I guess, as is the issue of biosecurity and what the role of understanding genomes, having genomes, can play in an improved notion of biosecurity. Just a couple of examples of some benefits, some direct benefits that have occurred. So we have medicines from nature, from plants. Here are some examples of some blockbuster, important medicines that have arisen from natural products. These, to be sure, were discovered and elaborated in a pre-genomic era, but the concept is that they come from genomes, and that nature really has invented so much over our 3.5 billion years of history, and it's embedded in genomes, and it's waiting for us to be able to unlock that information. So we have terrestrial plants, we have marine organisms, a group of vastly ignored species with respect to genome sequences right now, and some potent drugs that have come from those genomes. Fungi as well. Some very important drugs coming from there. And in that context, we do have kind of a concept that was developed and pioneered by one of the themes at the IGB, the mining microbial genome themes, headed by microbiology professor Bill Metcalf, that basically epitomizes this approach of sequence first and ask questions later. So the genome has the information, has the basis for that information. Let's sequence many, many genomes. Let's then use all the tools that we have in bioinformatics and in experimental biology, chemistry, chemical engineering, and so forth, to be able to then understand the molecules that are predicted to be encoded by the genomes and look for their biological activity. Switching from health to agriculture, it's sobering to realize that 12 species, 12 plant species, provide about 75% of the total food supply. And likewise, similar numbers of animal species for livestock and poultry production. So might there be new species for domestication, new gene variants to increase yield, new gene variants to increase environmental resilience, a topic of ever more importance. So the genomes are there for us to look at. For conservation, we have questions such as this: identifying genes for resiliency, climate change, mitigating effects of pollution and habitat encroachment, developing evidence-based conservation approaches, and I'll talk about one sampling method that we have in mind that relates directly to this point, and creating genomic resources to restore damaged ecosystems and perhaps even enable de-extinction. As I think many of you know, there is a project underway now to bring the woolly mammoth back based on a genomic approach and the tools of synthetic biology. There are many examples of knowledge that we've been able to obtain from genome sequences. I picked out a few here that I'll say just a word about. First, on the top right, we have the pioneering work of the Wright Project, led by Professor Steve Long in the Department of Crop Sciences and Plant Biology, that heads a large group at IGB to engineer photosynthesis. This is an image that was taken from their article in the New York Times a couple of years ago that showed an increase in photosynthetic efficiency of 15%. The last time double-digit increases in something related to yield was obtained was in the Green Revolution in the 60s and 70s. Since then, it's been single digits. So potential major transformative research going on that uses genomic information as a starting point, together with sophisticated modeling and plant biology, molecular biology, agronomy, engineering, to develop robots to obtain the phenotypic information. A really amazing project that starts with genomics. On the top left, we have a picture of chromosomes that are painted to reveal evolutionary relationships between distinct species, human and aardvark. This is the work of Professor Harris Lewin, showing that there are parts of chromosomes that one can relate one species to another, even if they're distantly related, and then look at structural similarities. In this case, Harris made the discovery of some parts of chromosomes that seem to be very fragile and prone to mutation. Changes, genetic changes that are associated with cancer. So having model systems as diverse as the aardvark to be able to understand the genetic basis of cancer opens up new doors for understanding the biology of cancer and then therefore possibilities for treatment. A little bit about work from my own laboratory and the honeybee, the lower left. We were able to use a genomics approach to be able to show that genes that are active in the bee brain of individuals that are relatively unresponsive to social information, the genes that characterize that social unresponsiveness, show interesting and provocative overlaps with genes that have been implicated in autism spectrum disorder. And I don't mean to suggest for a moment that these are autistic bees. Autism is a much more complicated syndrome than just that. The point is we can use genomics to create a building blocks model in the understanding of brain and behavior and look for what are those molecular roots of the social brain and then how are they used in different instantiations and independent evolutions of social behavior. Again, the genome at the foundation. And then finally, we don't have anyone working on the naked mole rat here on campus. I wish we did. An amazing species, and it's known as the perhaps only eusocial mammal. So that means it's almost as interesting as honeybees and ants and wasps and termites, and almost as interesting as not to do it justice. It lives an incredibly long life for a mammal, no signs of cancer. It feels little or no pain. And the genome has recently been sequenced, and people are really looking at the genome to see how does a mammal genome evolve such strikingly different traits. And obviously, I picked traits that have biomedical interest and importance. And so it's there. Okay, so that was a bit about the motivation. And in leaving, I will say, and perhaps seed a conversation, one can imagine, those of you that are biologists in the audience, thinking about, well, you know, there are limited resources, and should we learn more about the genomes that we have already, because we know so little about the genomes that we have, should we concentrate our resources on learning more about those genomes? They were carefully selected species to be sequenced as paradigmatic in many ways. Shouldn't we just kind of hunker down and really dive deeper and get that knowledge as opposed to sequencing everything? I hope we don't have to make that choice because the answer should be both, but that might be something for us to reflect upon. Okay, so what's our plan? Our plan is, the main approach will be to march through the tree of life. I have listed for you the main forms of classification of life on the planet, from the very highest levels to the lowest levels to the species level. And the plan is to take 3 of those levels, family, genus, and species, as our organizing forces. So phase 1 will be to sequence 1 representative of all the families. And for those of you not familiar with the nomenclature, I use an example of us as a species, and so we're in the family of the great apes. So there are about 9,000 families of eukaryotes on the planet, and the idea is to sequence 1 representative of each of those. You'll see the word reference underlined. This means that we call for the sequencing of these genomes to be at a very high level, high quality level. There are just 2 distinctions right now in this still frustrating era where we can't read the genome from the beginning to the middle to the end. So a reference quality genome is one where you throw everything you have at it, all the different technologies of genome sequencing, so that you have a genome sequence that can be resolved to the level of the chromosomes. We have 23 pairs, honeybees have 16, and so on and so forth. And so, so that you can have pieces of the genome that add up to the number of chromosomes you have. Chromosomes are a very useful level of biological organization, so that's considered the state of the art, reference quality genome. The other ones, cheaper versions, are draft genomes, contain lots of gaps, lots of missing pages, so to speak, if you like the book analogy, and have to be used in conjunction with a more complete reference quality genome to be able to be read thoroughly. So the idea is to sequence 1 representative of every family. And we estimate this to be about $500 million and to take a couple of years. Secondly, to the genus level, right? We're Homo sapiens, so to the genus level, and there are thought to be 140 to 200,000 known species of eukaryotes, known genera, of eukaryotes. So that's phase 2. And then phase 3, the roughly 1.5 million species would be phase 3. So that's our tree of life model. And that we think will be a dominant form to organize the project. We have a complementary approach that we've proposed, which is ecosystem sequencing, metagenomics in a more technical term. Go into a particular part of the world, either be chosen because it's a biodiversity hotspot, or because it's particularly endangered, or some other attribute that field biologists can offer up, and then go in, as I said before, and sequence first, ask questions later. Collect, sequence, and then collect that information. And this has some interesting advantages, in particular with respect to understanding how ecosystems are organized, how communities are organized, how different biological hotspots might be contributing to our understanding of biology in general. It can also help with respect to the fact that there are some 10 million species of eukaryotic species that are predicted to exist, and only 1.5 million have been described so far. So we expect that this approach would allow for species discovery as well as obtaining existing knowledge. And I note in the image that you have that there are a number of very high quality long-term observatories that have been set up, funded by various funding agencies such as NSF in their well-known NEON program. And so it could be very interesting to piggyback on those resources that already exist. So what will it cost? We estimate, based on current technology, that it will cost $4.7 billion over 10 years. To give you some perspective, the Human Genome Project, public Human Genome Project, which sequenced the equivalent of 1 genome, cost in today's dollars $5.9 billion. So seems like pretty good trade-off there, 1.5 million species compared to 1, but that's just me talking. So that's what we note here. Return on investment, the Battelle report very authoritative analysis on the impact of the Human Genome Project, reported a return on investment, an ROI of 65 to 1. And again, for one species, we might predict confidently an ROI even greater for the Earth BioGenome Project. So that's a little bit about the estimated cost. How about the technology? Well, the estimates that we've generated and the timelines we've generated are based on existing technology. In fact, not even the latest technology. The experts in the room will know that this is a machine from the company Illumina, which represents the second generation of technology, not the third generation. And so we're basing the estimates just on very known genome sequencing technology for this project. We are going to need a lot of help in this project from our computing colleagues. So this is an analysis we did a couple of years ago. It was a collaboration of a number of labs, Ravi Iyer's lab in ECE, Saurabh Sinha's lab in computer science. I contributed a little bit the cutesy title of the paper. That was about the limits of my contribution to this project. And I will say it's the biggest failure of the project because no one has dropped the word astronomical in favor of genomical yet, as far as I know. So, but the point is that doing very careful analyses and projections of the needs of computing needs of genomics, they dwarf these other three big data domains. And to put that in the context of the Earth BioGenome Project, we need to telegraph that this is what's coming because there are no bioinformatics tools that are computer programs that are used to analyze genomes. There are no bioinformatics tools that can scale to the level of what we're talking about. So it's not a matter of taking an existing tool and tweaking it to be able to work better for a larger number. There aren't any that'll work for this large number. So part of the motivation of the project is to put the computing community on notice ahead of time that this is coming down the pike and we really need your help to build the tools because genomics can only travel as far as computer science and computer engineering will allow it to. So here is a roadmap that has been developed for the project. Specimen collection. GGBN is an organization, an international organization that already has been involved in collecting specimens and determining the right kind of storage conditions and how they match with the technology needs for DNA sequencing. So we have the project phased, specimen collection, sequencing, analysis, and annotation. So those words there, as I said, you take the genome, chop it up into lots of pieces. The next step after that is you assemble the genome. Then you annotate the genome, which is you try to identify as many genes in the genome that you can recognize on the basis of their similarity to genes that already have been identified. And from that, you can start to begin analyzing the genome. How many genes are there in a particular family of detoxifying enzymes? Has it expanded? Has it contracted? How does that relate to another species that may or may not have been known to be related to it or not, and so on and so forth. So that's the process of analysis. So probably those two words could have been reversed, annotation and then analysis. So we are working on the project. We have a large working group, an international group of excellent, diverse scientists from around the world. And the working group is working. So I'm showing an org chart, not to bore you to tears, but I want to talk about two subcommittees to highlight some of the difficulties associated with this project. So really at the end of the day, and I've kind of been saying it, the idea of sequencing the genomes and even doing the rudimentary analyses on it, yes, we need those new tools, but we've been there, done that. We know what those tools are supposed to do. That's the easy part of the project. The sequencing technology is there already. We know in outline what kind of analyses need to be done to build the rudimentary library of knowledge here of genome sequences. The hard parts are actually the front end and the back end. So what do I mean by that? The front end, the subcommittee for sample collection and processing. There may be sensitive parts of the world that are very difficult to reach due to a variety of physical or political reasons. And so we imagine that the project's going to require innovation in sample collecting, sample identification, sample sequencing. So here are some slides here of existing, you recognize this, these technologies, of course, a drone to be able to move remotely, and then an iPhone, and then a tiny little sequencer that's the size of a thumb drive. And so we might imagine that it's going to be necessary to program drones to be able to collect specimens in certain parts of the world. We might imagine that we want to develop, and by we, I mean the systematists in the community, developing tools to be able to identify species in remote areas, maybe to access databases and so on and so forth. And a lot of that's already starting to happen. And we might imagine sequencing on location for certain kinds of situations. What kind of situations might they be? Well, that relates to the next subcommittee to highlight, and that is the ethical, legal, and social issues associated with the project, having to do with the Nagoya protocols and access and benefit sharing. So I know many of you out there do field work. Some of you work internationally, and you know that the climate has changed appreciably in some parts of the world with respect to the ability to obtain biological material and take it out of the country. And so basically the narrative is that countries increasingly appreciate the value of their biological resources. They look back upon what happened in the 19th and 20th centuries, and they say, nah, we're not going to do the, give away our resources like rubber, like we used to. This is our material. We want to keep it in country. And so how do we square that with the goals of having this be an open access project where the knowledge is shared across the planet? We don't know yet. There are some really delicate sort of geopolitical issues related to this project. We have a stellar subcommittee headed by Melissa Goldstein, who was did LC work for the Human Genome Project. She's a professor at George Washington University, and she was in the Obama administration OSTP, Office of Science, Technology, Policy, and she's putting together a group to help give us some guidance on how we're going to do this. And that's another topic that, especially in a diverse group like we have today, we might want to talk about a little bit. So how does one guarantee open access to the information in a climate when countries are becoming increasingly protective of their resources? So the way the project is looking then is it's a network of networks. So many of you know that there are genome sequencing projects going on. The I5K sequence 5,000 insect genomes that I started a few years ago with Kevin Hackett. Vertebrate Genome Project, a large-scale plant, 10,000 plant genome project. So things are going on. And there, the way we interact, we being Earth BioGenome Project, is first get out of their way, not interfere with anything that they're doing, but talk about standards for genome sequencing, as I mentioned to you before, so that at the end of the day, we do have a, quote, library of genome sequences that are similar, so that people can go across these different domains of species and get large-scale synthetic information. So we wanted to do that. We also envision increasingly national projects, as I'll show you, projects that are originating from countries rather than defined by particular taxonomic groups. So that's the plan. What's our progress so far? We have 26 and counting institutions that have signed an MOU that just say they support the project. No money is committed there. So you see Illinois is on there. So my colleagues, don't worry, I'm not taking any money away from you. It's just a statement, an affirmative statement of support for the project. And we have an increasing number of institutions that are signing that MOU. There are 24 and counting affiliated community, regional, and national projects. By community, that means sort of the biological community, Fungal Genome Project, for example, I5K, as I said. We also see regional and national projects shaping up. So the first regional project is a project that was approved by the California legislature for a California EBP project. There are a number of companies that have signed on. So in the connection of the bioeconomy, so we have a set of companies led by the CEO of Illumina, the largest manufacturer of genome sequencing equipment. They presented this letter to the OSTP in support of the Earth BioGenome Project. Our biggest success so far comes from the UK, and we had the formal launch of the project last November 1st, 2018. The Wellcome Trust, the largest philanthropy in the UK, has agreed to sequence all the species in the UK, some 66,000 known species. They've committed a large sum of money. There's a separate, so it's an EBP project, but it's got its own name, the Tree of Life Project. This is going to be centered at the Sanger Center in London. And Mark Blexner, a well-known geneticist from Edinburgh, was recruited to move to the Sanger Center in London. They're staffing up a whole program. They're talking about how to collect the species. They've got a timetable, and we're just absolutely thrilled with that first project. We also note that it has nothing to do with our plan of marching through the Tree of Life in this global sense. And I will admit that the three of us were a little naive about the times that we live in when we proposed this sort of post-Star Wars intergalactic, let's just, sing Kumbaya and march through the Tree of Life together. Rather, we're seeing, in this case, a funding agency, philanthropy, saying, yeah, we'll do it for the UK. And when we say, hey, how about, the rest of Europe? Nope. We're going to do it for the UK. And we have a number of other projects that are in the pipeline that have that approach. And it poses some interesting challenges because I think many of you might realize that a species that's in the UK might also be, say, in Sweden, a couple hundred miles away. So how are we going to handle that? We don't know. There are some possibilities. We could say let 1,000 flowers bloom and let the countries sequence all of their species, and we might learn something from geographical differences and so forth. Or we might think of more top-down integration or meddling, depending on your perspective. So we're just, kind of dealing with that. But I would say having this commitment is what is classically called a good problem. And so we're happy to deal with it. So that's the first bullet point there. The launch, I'll give you the quote from Nature and the Times of London when I wrap up. Other projects, national projects, are in planning. I will also mention that one challenge of this project that we did not anticipate is the changing political climate in this country with respect to relationships with China. China is a world leader in genomics. They've committed huge resources to genomics. They have a really large and very high quality infrastructure. And when you mention the word China in the current OSTP, that's not an easy conversation. So again, how do we juggle the fact that this needs to be a global project with open access, with a change, a stark change in the political environment across the world? I will note with candor that we don't yet have a comprehensive national project in the US. We have a small project in the USDA to sequence 100 of the top insect pests and some conversations, but no large-scale project. That's what we're hoping OSTP will help catalyze. For those of you that are familiar with the BRAIN initiative, OSTP under the previous administration played a very important convening role. The Office of Science and Technology Policy does not have money for grants and large-scale initiatives. What they have is a convening power. if they say, hey, we're going to talk about the BRAIN initiative and what all of you federal agencies, some 25 agencies, NSF, DOD, NIH, and so forth, what you're doing about it, can you come and show, can you show up Monday morning at 9? You will get them to show up Monday morning at 9. So there's a convening power. And what happened in the BRAIN initiative was that it socialized the federal agencies to the importance of the project. They did as what agencies always do. They said, oh, we're already working on it. And here's our part of our budget that is actually BRAIN initiative type work. And so we're going to call that BRAIN initiative research, and that's fine because that gave the project momentum. And then when new funds were available, whether it was because of a political opportunity or some kind of moment in Congress, the project was there waiting to go and then did get additional funding. So we hope we're at the beginning of that process where OSTP can socialize the project in the context of a larger interest in the bioeconomy to get federal agencies to ask, at least themselves, how might they see themselves contributing to the project. Another sign of progress, the Vertebrate Genome Project, and another example of get out of their way and let them do their work, released 100 reference quality genomes for various groups of vertebrates that you see here. And that was a cooperative project with the groups that you see on the right, generally under the Earth BioGenome Project, but started long before the Earth BioGenome Project started. And then finally, this very wordy slide that indicates how OSTP is seeing the Earth BioGenome Project so far, and is not coming out with a specific endorsement of the Earth BioGenome Project, but after we proposed this, it led them to realize that, by contrast to several other nations, including China, we do not have a global strategy for genome sequencing. And so what might that global strategy look like? And we've proposed one way that it might, one model that it might look like, but the idea is just to put that on the table. So that's the Earth BioGenome Project. We want to create, I think, a digital genome repository of life on Earth. We think that it'll provide the infrastructure for a new revolution in biology for the reasons that I said. If you want to hear more about it, read more about it. We published a sort of a manifesto last year on the project in PNAS. I'll be happy to send it to you if you'd like. And I'll close with just a couple of quotes. This was an editorial from Nature that endorsed the project after the launch in London last year, excuse me. And this is an even pithier quote from the Times of London. So we like those 4 A's, and everyone likes to get an A, right? So we got 4 A's. And happy to take your questions. I want to acknowledge graphic design of these slides, Nick Vasi, IGB Director of Communications and Engagement. Everyone in the Earth BioGenome Project is now using these slides, this design of these slides, even though this is not an IGB project per se. So thank you, Nick, if you're out there, and thank you for your attention. I'll be happy to take questions.
Imagine how you sequence an individual. How do you sequence a species?
So that was like a shorthand. So an individual that belongs to a species. So we get
variability that you're not capturing.
Correct. That's right. Yeah. So this is just the beginning, and there's a whole science of population genomics to do just what you're saying: collect multiple individuals from the same species to look at the variation and how it relates to climate, to geography, to behavior, and so forth. So you're exactly right. But this is just the bare bones. Yes?
Thank you. That's I've lost my voice this week teaching. That's exciting stuff. Let's see if I can do this. To what extent is this project kind of an updated version of what is called like salvage anthropology or salvage ethnography? The way, the way that people in the 19th and 20th century basically got all the information they could about cultures before they disappeared in zoology and in biology. So, I mean, you began with slides that show the sixth extinction and all of that. Could you say a little bit more about the relationship between the disappearing of life? Excuse me, what is embarrassing?
You must be teaching well this week.
No, I
David?
don't teach tomorrow. I don't sound like this. And the and the goals of the project. How do you align a project like this with, say, Ed Wilson's desire to actually, what he wants to do is say, let's identify everything before we, before things disappear.
Yeah, no, those are great questions. Yeah, the question was how do we align this project with other large-scale projects that have to do with capturing elements of knowledge related to what exists on the planet before it goes away. So it was mentioned Ed Wilson and some of his ideas. He's a member of our advisory group, and when I called him to ask for his support, he said, well, Gina, I'll support this project, but I would be more supportive if you did the other 8.5 million species that are undescribed. I said, thanks a lot, but it speaks to the issue. So, all of these efforts are in part motivated by the fact that things are happening fast, that we are losing our biodiversity, and one idea is that at least if we have the genomes, then that can provide some of the knowledge that exists. It's no replacement. We don't mean to endorse this as an idea instead of the rest of what's required for conservation, but it's one part of it. Yeah.
So as you go forward in your project, a substantial portion is dedicated towards identification of new species. And when that comes around, say, in reference to the first question, what are you going to use as a species definition?
Yeah, great question. Well, we have systematists and other evolutionary biologists that are going to be on the working group, and we'll deal with that. So I don't know whether, and I'm sure I know we have experts in the room. Should there be a change in how that's probably a dangerous question to ask at 5 minutes to 5. Should we have a change in our species definition? But it's a fair question. And so I'll frame it in another way. Right now, one of the most important concepts in biology is the concept of orthology. The idea that a gene in species X, and you've sequenced the genome, you know nothing about it. The first thing you do is you ask, have we ever seen any gene similar to that gene in question in any other species? And if it's similar enough in sequence for a variety of very interesting biological reasons, you can say, yeah, it's similar, and it's probably a cousin to that species. Every time a new genome is sequenced, there are 10 to 20 to 30% of the genes come out as novel, never before seen. Is that because we've only sequenced the genomes of 0.3% of the species, or is it because species have their own sets of genes? And if they do, how does that relate to the question that you're asking? If they don't, then we need to fill out the tree of life and get better understandings of orthology relationships. That would also have a different implication. Notice I didn't answer your question because I have no idea. Yeah.
Thank you for your presentation. I wanted to see if you could expand a little bit on something I noticed. I feel like two motivations of this project is the bioeconomy and biological conservation. And those motivations can sometimes be in direct conflict of each other. And I also noticed you have about 20 corporate partners, but I didn't see any partners listed, environmental conservation organizations or anything else. And I'm just wondering how you're thinking about that or rectifying that what could be sort of a paradox.
Mm-hmm. Yeah, no, that's a great question, Nicole. Natalie, sorry. We have other partners that are actually the California project. It was advanced through the legislature by a conservation organization. So I showed that particular group with respect to the bioeconomy side of things. But we have to have both. There's a large philanthropy in California that's very conservation oriented that we've been talking about a project with. They're related to the Restore project in California. And so they're very interested in this from the conservation perspective, and we're going to definitely have full representation and have to balance those. Yeah. Mark?
Well, there was a report recently that the U.S. museums or museums in general have a male bias.
And so I was wondering, what is your gender genomics policy? We're going to sequence males, females. In some species it makes sense. In some species it doesn't. How's that going to work out?
Yeah, that's interesting. I hadn't heard that result, and we'll make sure that we address that. Thank you very much. Yes, in the back. Yeah, Esther?
So I realized also that I can see a lot of representation of African continent, yet a lot of human life and past life is attributed to the continent. And I know that the continent doesn't have huge resources. So how do we balance between countries that have resources and the ones that don't have resources and share this sort of technology?
Yeah, so I didn't really draw out the problem sufficiently. Thank you for adding that part. So most of the Earth's biodiversity is located in developing countries. And the question then holds, as you said. We are thinking about some ideas, so see what you think about this. One possibility is to get sequencing companies to donate equipment to create sequencing labs in country and provide training, both species identification, so parataxonomists, as well as sequencing technicians to be able to sequence species in country and then therefore keep the biological material in country and then deal more specifically with the digital information, which is also a big deal, but to deal specifically with that and grow new sequencing communities, sequencing-based communities, academic communities that have that knowledge in those countries. So that's one idea that we hope to be able to do. We're in discussions with the Gates Foundation about that. They, as you know, have a very strong interest in Africa. And from the point, the argument that we're framing to sequencing companies that you could be growing your next markets, creating your next markets in these countries if you empower people to be able to appreciate genomics and be able to use them. But you do amazing work in a variety of countries. We'd love to get your perspectives on it. If you want to be on a subcommittee, we'd love to have you. We really need that. Yeah.
So a lot of existing natural history collections have a lot of these specimens already. They're already identified. A lot of recent literature shows that they still contain viable DNA.
Mm-hmm.
To what extent do you think those resources can be used for this project?
Yeah, so we're definitely keeping track of that. So Smithsonian, for example, has a leading role in that area, and they have collected material for the lab, you know better than I do, for the last 10 years actually in liquid nitrogen. They have an off-site facility with just a whole warehouse full of liquid nitrogen containers. The issue has to do, did everyone hear the question about collecting? The issue has to do with the compatibility of the storage medium and the technology. So I mentioned the Neanderthal story. You can get degraded DNA, sequence small fragments of it, put together a genome painstakingly that way using second generation technology. That's Illumina. Third generation, where you have longer reads, you need high molecular weight. You need bigger pieces of DNA, not compatible with some of the forms of storage. So it would be great if we could use both pinned, frozen, even alcohol preserved specimens for some cases, and then all of this liquid nitrogen stuff. But the sobering reality is that for the latest technology, that won't work. So it's going to have to be some kind of a mixture. Yes.
It's related question about voucher specimens because you need to think about depositing those voucher specimens which you sequencing in national traditional collections. And my question is how much money you are committed from your kickoff plans to support national collections in different countries to store voucher specimens for your project?
Yeah, so the question was how much of that estimate, 4.7 billion, goes towards specimen acquisition and then proper storage. We've consulted with experts on this from the Smithsonian, from other museums, and they've given us a rough figure for what that is. I don't know what that is, but we have considered that very carefully. And I'd be happy to put you in touch with those individuals if you had any particular thoughts on that. getting to be 5 o'clock, and I want to be mindful of some people needing to leave. So maybe I'll take one more question. I'll be happy to stay after and talk with whoever would like to. So Mark had his hand up for a while. Mark?
Yeah, you at first mentioned a clear separation between the BioGenome Project and the bacterium archaeal projects, called sequencing. But today, I think we all know, or it's accepted, that we're actually superorganisms, right? Is there a plan to, as you sequence a species, to also sequence its synergistic microbiomes?
Yeah, that's a great question. We hope so. So this will play out. This is a decentralized project. The Darwin Project does plan to do microbiomes. They're hiring people related to that aspect of the project as well. And so if that's a pace setter, that's that's a sign of good things to come.
Thank you so much, Jim.